Hello, good morning, everyone. Thank you for coming, and hello to everyone online. I will start my talk going straight into how, in a very large consortium funded by the EU, we have addressed everything we could about this disease with, to my knowledge, every possible approach that science has at hand. And unlike many other similar consortia and initiatives, rather than using one or two techniques, we use them all at the same time. Quantitative techniques to try to understand if there's any biological mechanism that would be the leading cause of this condition. What is the role of a virus in potentially causing the symptoms? What changes are happening in the body? What do those changes tell us? And could they inform us about how to define these conditions better and how to possibly intervene? So I will give you a glimpse of mechanistic. I cannot show them all because it would take days, an infinite amount of graphs. I spare you that. But I'll rather take few examples and give some hopefully positive take-home messages. And then I will leave the floor to my colleague Velina, who will move into more into patients' studies. So before going into long COVID, I will just to remind you that to virologists like me, the fact that viral infection could manifest into long-lasting symptoms was not new. Of course, the pandemic brought all of us into the spotlight, which was good and bad for other reasons. But all of us were fully aware that in a number of people, a virus infection can leave a tail of long-lasting effects. And I don't know if you have followed the last 2-3 years of this type of research that links a virus to a disease that becomes chronic. But thanks to projects like this, even bigger than this, for example, we now know that multiple sclerosis is linked to a virus. Can you imagine? If we would eliminate by a vaccine Epstein-Barr virus, multiple sclerosis would disappear. That's how powerful this type of studies are. But it took decades and 10 million US soldiers to be studied across 10 years to finally pinpoint that you need to have one virus to then have that disease. And even then, we still don't know why some people have that virus and don't have multiple sclerosis, and then other ones do. So there are other factors. Multifactorial conditions are essentially probably most, if not all, of the chronic diseases. Similarly, long influenza, long measles, long COVID, these are well-documented conditions. They share multiple features, not all, and these features are different from one person to another, which makes it extremely difficult to define them and to understand them. But all of them have, in my opinion, I'm a virologist, so to me everything is a bit more virus-centered than perhaps a person that studies bacterial cancer. But many of them, let's put it this way, they have infections, long-lasting inflammation. Even neurological diseases such as dementia and Alzheimer now start to be linked to herpes viruses, such that people who have vaccines, they have up to 1/4 less probability to have those diseases. And we know near nothing about the mechanism of how a virus such as a herpes virus or any of these and many more could actually link to long-lasting conditions, which is what my lab is interested in. So in our consortium, as I was mentioning at the beginning, we decided to take a very comprehensive approach that starts from, let's see if I have a pointer here. Yeah. Cohort studies. So we start with patients, of course, as usual. People who have severe symptoms, You can imagine from a scientific point of view, starting from people that have very strong symptoms, it's a bit easier because the symptoms are more defined. Then controls, people who are healthy, or we consider them healthy, because I would like to inform you all that the idea of a non-infected person doesn't exist. All of you, at any given time in your life, have tens of viruses in your body. We could define it as the virome, similar to the microbiome that you all are familiar with, probiotics that are part of you and are extremely important for you. At the same time, you also have a number of viruses. We still don't know which one is good and which one is not, but there is no non-infected person. You can imagine how this complicates studies when you're comparing a person that has one virus with a person that doesn't have it but has many other viruses. How do you compare things? Then, very comprehensive characterization of these patients in the clinic. Elena, the leader here. Mechanistic studies, starting from a single cell, a single neuron, which is what we do in my lab, to mouse models, to organoids. We can grow tissues in the lab in 3 dimensions and try to understand everything we can about, can a virus like SARS-CoV-2 infect neurons? How much? How fast? What happens to a neuron when it's infected? How does this compare to other neurotropic viruses? What happens to the viruses that are in the body of a person that has been infected with SARS-CoV-2, the causative agents of COVID-19? Does the virus evolve inside the person? Does it mutate over time? Are there specific mutants that correlate with long COVID? Not really. Doesn't seem to be the case. Your antibodies, your T cells, B cells, what is happening to them? Your immune system. Coagulation factor. How is your ability to cope with lesions and potential hemorrhagic events? How is it affected by an infection? And then mechanistic studies where we take pluripotent stem cells, we can differentiate them into neurons, glia, any other tissue of the body, and systematically and quantitatively assess whether this particular virus or any variant of it has the potential to infect neurons and the brain. Then the hope to define with specific biomarkers what this disease is. Is it different enough from other conditions such that we can have measurable parameters that we can use to detect it with a machine, with a tool, maybe with a test? So multi-omics analysis, sequencing of entire genome across thousands of people, proteomics of the entire blood, lipidomics, immunological characterization, even the microbiome, even the bacteria from the gut, to see if there is any change whatsoever. Metabolomic studies, I will show you a bit about this. And then interventional approaches. Everything we can do in real time to see what treatment can help people, how many do recover, and those that don't, how is their body behaving. This across a period of time that it's a race against the clock, because as the vaccines has slowly but surely decreased the hospitalization of patients, then you have less and less patients, and therefore those that you have, you cannot follow them for a very long time, which makes it very difficult to study. So today, my colleague Velina will talk to you about the cohort studies, the interventions in the clinical settings, whereas I will tell you more about the mechanistic studies. And as I said, it would be impossible to present all of this in any understandable form, even to myself, to be honest. But I will try to give you take-home messages that, in my opinion, accurately summarize what we have found so far. So I will start with the mechanistic studies to address one of the things I'm asked the most. Can this virus infect the brain? Well, if it can, the first thing it should do is to infect neurons or cells of the brain. And that I can test. In the laboratory, we have essentially every human virus that you can imagine, including the neurotropic viruses. So we know what a neurotropic virus can do when you infect neurons. And then we can compare the SARS-CoV-2, every variant, with those that are well-known neurotropic viruses. And the results are this. We can very easily detect and infect the neurons. These are images from mini brain that we can create from stem cells in the lab, so no animals are needed here. We differentiate the cells so that they become neurons and glia. You can see the neurons in red, and when they are infected, they are highly visible because the neurons become fluorescent. So with robotized microscope, we can immediately see if a neuron is infected or not, if it changes shape, if it doesn't work anymore, everything you can imagine. And this is a very clear summary of what I'm trying to deliver today. SARS-CoV-2 really is not a neurotropic virus. It's a very good respiratory virus. It causes a lot of symptoms in the body. It will continue to evolve. It will stay with us forever, but it is not a neurotropic virus. You can see that the amount of neurons that in a laboratory we are able to infect, meaning I can put as much virus as I want into those neurons, even an exaggerated amount that you will never encounter in your life, and still 0.01% of cells would ever get infected, and they will die after a few days. If you do the same experiment with any known neurotropic virus, Zika virus, encephalitic viruses, herpes, tropical viruses delivered by mosquito, you will infect them all in 2 days. So this, our laboratory and hundreds of laboratories in the world have tried to reproduce almost with the hope that they would infect it, because if you're a virologist, you sometimes hope that you will see something and you can study it, but this virus is not really good at infecting neurons, probably because the receptors are missing. That's one of the main reasons. And so to see the association between an infection that a person can have and the symptoms that can develop, we approached in this very broad manner, measuring everything that it is possible to measure, from the blood, from the stools, even from the cerebrospinal fluid. And this is what I would like to show you today. So as an example, I would like to introduce you to an analysis that it's being currently finalized, and Alona is here sitting. If anyone has technical questions, she'll be happy to answer. Essentially, a patient that had very strong neurological symptoms were hospitalized, where they gave consent to donate a bit of their cerebrospinal fluid, and similarly, patients that did not have those symptoms also donated those samples, and we could analyze all the metabolites that are available. And to give you a very short introduction, why are we interested into using metabolomics as a readout for the status of a patient? So the metabolome is, you have to consider it as a biochemical reaction that are happening in the body at all times, and they are in charge of functions and resilience. So in other words, it's like a seismographer for an earthquake. When you measure all these parameters, you have a very accurate picture of how healthy or how distressed that body is. They're highly dynamic, and it's the first buffer that your body has against exogenous stress. So when some values go up, other ones will go down, try to find an equilibrium. If something is missing in your body, something else will be produced to keep you always in the balance. These are thousands of compounds that reach a constant fluid state, and nowadays with mass spectrometry, we can analyze them all at once in every patient. And so we thought that wouldn't it be sensible to take a sample from the liquid that usually irrorates your brain because if something would change in the brain, sure enough, we would see it from there. And so taking samples from the spinal cord, the blue fluid is the cerebrospinal fluid, all right? So samples are taken from the cerebrospinal fluid and from the serum, like from the blood, as a comparison. Then they're passed through mass spectrometry, and then the data are analyzed. And of course, you get thousands of results, but you can organize them so that they're very easily understandable visually. And so, for example, you can take 14 patients that have very strong neurological symptoms and 11 patients that have neurological symptoms of long COVID and 14 control patients. And for each of these, you can analyze hundreds and thousands of compounds in the cerebrospinal fluid. And in this heat map, the values that go below the threshold, the normal values, will be blue, and those that are going up more than normally, they are red. And so if you take a bunch of these results, the one that changed the most, in your control, you can see that normally, for example, these molecules here are more or less very highly expressed, whereas this one are expressed at low levels. But if you measure the same in the long COVID patients, you can see that this pattern is inverted, to give you the feeling that there is no problem distinguishing between a person that has a strong condition and one that doesn't. The challenge will then be to interpret this and to understand what they mean and how can we use it to help those patients. So, for example, a general take-home message for this type of analysis is that the key observations are that the patients have signatures of decreased brain energy metabolites that indicate low energy metabolism in the brain. And now we know that this virus is not really good at infecting the brain, so this would suggest to us that there is something happening at the systemic level. By analyzing each of these compounds, the expert would tell you that there is oxidative stress signatures characterized by reduced glutathione levels, for example, is one of the molecules. And this is consistent with findings from other labs in the world. I give you the references here. But now, the important notes that we must always do when we do this kind of analysis. The metabolic changes that we observe can be a cause of a condition or the consequence of the condition. And in this type of experiment, we cannot distinguish. We can just record them. It will then be follow-up studies and intervention studies that will allow you to know in retrospect if something was the consequence of the cause. In addition, every patient has a history, a medical history, and that medical history has to be taken into account. So the comorbidities or other factors such as age, their global health, their stress levels, everything that some people consider psychosomatic in the scientific world is not called that way, because everything you go through in life will cause a biochemical change in your body. So even when people disagree with the word, for us, nothing is sort of superficial. Everything is quantitative, and it will cause a real effect, a real change in the hormonal and chemical level in your body. And therefore, once we get these results, it's the beginning. Then we have to see whether what we found, does it define long COVID, or is it the consequence of other things that these patients have in common? And as you can imagine, the problem is that if you want to follow patients, it takes time. So the answer that I would like to give people every week don't come in a week or in a year. You will have to follow them for decades. And hopefully this is what we all are doing at the moment. So to conclude, after approaching essentially this problem with every tool and weapon we had at our availability, the conclusion is, in my personal opinion, at least I take this responsibility, is this: the long COVID cannot be defined by a single biomarker. So don't expect that you will find one molecule that you measure in your body and you know if you have long COVID or not. It doesn't work that way. It's a much more complex multifactorial disease, and it's not even a new type of condition. We have found, and many other colleagues have found, some genetic risk factors that make some people more prone to develop these conditions, but long COVID is not associated with a specific human gene. So it's not an inheritable genetic disease. That's also been shown convincingly. Then multi-omic analysis can easily distinguish between long COVID patients and so-called controls, but the effect of comorbidities and other factors needs to be studied further and across longitudinal studies. And even if the mechanism is not fully clear, I think this is my positive take-home message, and Velina will show you a bit more in practice what this means. Even if we don't understand every atom about the condition, this doesn't mean we cannot intervene to help. In fact, I will tell you that for the vast majority of our diseases, we don't know much, and we still have found a way to intervene. So this means that, yes, we will continue to try to understand everything we can possibly understand of these conditions, but meanwhile, people are getting better, many of them, and those that don't become our focus for the future. So I would like to thank you for your attention, and if there's questions, of course, I will take them. If not, I will then let Velina continue with more inpatient approaches. Thank you.
Maybe we have time for one or two quick questions, or maybe we can go forward. Yes, one question.
Same. I will repeat the question. Go ahead. So the question was that if you perform these kind of studies, let's say a metabolomic study, lipidomics, proteomic, whatever it is, do you find similar results for other conditions, or are these results specific for the long COVID, essentially? And the answer is, some of the results seem to be specific for the patients that are in the clinic with long COVID, and some are very much shared by many other diseases. But this is different from one patient to another. So far, there's no consensus of one type of change that would only be associated with the long COVID, number one. Second part of the answer is, when we then dig in and ask what kind of medical history those people have, you may find that the same changes are equally associated with a number of conditions that they share. So which one is it? Is it the condition they already had that made them more prone to develop, let's say, a more serious symptoms, right? And the two things combined, or is it one or the other? And these things require, unfortunately, an incredible degree of analysis time. We're even using AI to try to untangle, all these parameters to see if there are strong associations. So, and this is very much ongoing.